Good evening, one and all, and today we are going to discuss about oligosaccharides. What is oligosaccharide? Oligo means few, saccharide means sugar. So oligosaccharides are those sugars which are basically having those carbohydrates which are have made up of more than one one more than one monosaccharide units. Monosaccharide units are simple sugars which constitute together to form oligosaccharides. On hydrolysis, oligosaccharides produce or yield uh, 2 to 10 monosaccharide units. So they yield 2 to 10 monosaccharide units. These are less soluble in water. These are less soluble in water, as well as these are having uh, less taste, less color, less whitish color, as well as these are having less taste, less whitish color, as well as um, these oligosaccharides are joined together by the monosaccharides are joined together to form oligosaccharides by the glycosidic linkage, and there are types of glycosidic linkage as well, which we're going to further study on the basis of the structure of particular monosaccharide as well. So here, basically, what we are having is that inside the oligosaccharide, there's less structure, there's less taste, and they are less soluble in water. And what is the general formula? The general formula is C12H22O11. So here these are divided into their types depending on the number of monosaccharide units. For instance, we are having disaccharides, we are having trisaccharides, we are having trisaccharides, disaccharides, tetrasaccharides, pentasaccharides, hexasaccharides, and so on. But most commonly, we are having disaccharides. Disaccharides is the most common oligosaccharides in carbohydrates, and these disaccharides are mostly common, and these are the ones which we are going to study right now. After which there are trisaccharides, which are also present here, which involves some examples of um, uh, for verbacose, which was trisaccharide. There are tetrasaccharides as well as pentasaccharides, but we are gonna specifically study about disaccharides here, because structure of a disaccharide can always other structures such as tetrasaccharides, trisaccharides are dependent upon the structure of disaccharides. If you're going to understand the structure of disaccharides, you will be able to understand other structures ultimately as well. So here disaccharides are having of are basically the most common type of oligosaccharides, and these are mostly having three major types, which include sucrose, fructose, it's sucrose, it's maltose, and lactose. First, we are going to discuss about sucrose. Sucrose, also known as cane sugar or table sugar, is a sugar which is a kind of non-reducing sugar, which means it cannot be reduced or oxidized or cannot undergo chemical reaction, due to which it is also uh, considered as an ideal transport sugar for translocation of food from one part of the plant to another part of the plant. So as this sugar is as translocation of food occurs from one part of the plant to other part of the plant, there is no need of chemical reaction or metabolic reaction, which should not occur inside the particular uh, lumen, inside the particular space of phloem. Therefore, sucrose is a non-reducing sugar which does not undergo any sort of chemical reaction, neither oxidation nor reduction, due to which it is considered to be an ideal transport sugar during translocation of food in plants, and that is why it is also known as transport disaccharide. So, afterwards, what is it actually made up of? It is made up of alpha D glucose. D represents dextrorotatory glucose, which means that in dextrorotatory glucose, the the hydroxyl group on the second last carbon is present on the right side, and in levorotatory glucose, the second last carbon is present on the left side. Accordingly, why is it known as alpha? Alpha is there because the hydroxyl group is present on the downward side, and beta is there because of the hydroxyl group is present on the upward side on the first carbon. So I hope this sounds clear. So alpha D glucose and beta D fructose are combined together in such a manner that C1 of alpha D glucose, the hydroxyl group of carbon 1 of alpha D glucose reacts with the hydroxyl group of C4 of beta D glucose, which collectively forms um, a glycosidic linkage, which is in actual the condensation process in which the glycosidic linkage occurs, due to which a water molecule is liberated and a new uh, compound is formed, which is known as a disaccharide. And that disaccharide, which is formed here, is known as alpha D1 alpha D alpha D1 beta D1. Sorry, it's alpha D1 beta D2 glycosidic linkage. uh, One problem was there that unfortunately it's in alpha D glucose there's first carbon which is giving its hydroxyl group, and in beta D glucose there's second carbon which is giving its hydroxyl group. So it is alpha 1 beta 1 glycosidic linkage, or alpha D1 beta D1, sorry, it's alpha 1 beta 2 glycosidic linkage, or alpha D1 beta D2 glycosidic linkage accordingly. So this is how the structure is formed. And one more thing, that it is also utilized as artificial sweetener inside our food. So this is a structure. uh, One more thing about non-reducing sugar is that sucrose and all of the polysaccharides are non-reducing sugar, whereas the lactose and maltose alongside all of the monosaccharides are reducing sugar, which means they themselves undergo oxidation and cause other materials to become reduced while without hydrolysis. So these undergo oxidation themselves, but cause other materials to become reduced by the process without hydrolysis. So they undergo oxidation without and cause other to become reduced without hydrolysis. So these are those sugar reducing sugar. And now we are going to move forward towards the reducing sugar, which is maltose. Maltose is also known as malt sugar. Maltose also known as malt sugar. uh, this malt sugar is an intermediate disaccharide between the starch and glycogen. Sorry, not the starch and glycogen. It is an intermediate sugar between starch and glucose. So first, when starch is broken down, it is broken down with the action of amylase into maltose, and then maltose is then converted by the enzyme maltase inside into the glucose. Accordingly, then the digestion occurs. Accordingly, the respiration occurs in order to gain ATP from the glucose. So here maltase enzyme digests the maltose, and here maltose sugar is found in germinating seeds. Germinating seeds or young seeds. Germinating young seeds. It is found in germinating or young seeds. Young seeds, and it is basically made up of germinating young seeds, as well as it is basically having what? It is having two units. First unit is alpha D glucose, and second unit is alpha D glucose as well. So it is made up of two units of alpha D glucose, out of which the long bond which is formed between both of the alpha D glucose is in such a manner that C1 of first alpha D glucose, having its hydroxyl group, uh, reacts with C4 of second alpha D glucose hydroxyl group, so that both of the hydroxyl group combine together to form a water molecule which is liberated, and they form a bond which is known as alpha D1 beta. No, there's no beta linkage, so we'll call it as alpha D1 4 glycosidic linkage. Ultimately, then it will be the bond which is alpha D1 4 glycosidic linkage, alpha D1 4 glycosidic linkage, after which this maltose sugar is basically having um maltose sugar is also considered to be a sugar which is found in the downward parts of plant. Finally, we are having the lactose sugar. Lactose sugar is made up of, uh, also known as milk sugar, is made up of beta D galactose and beta D glucose. It is made up of beta D galactose and beta D glucose. So here, here the lactose sugar is made up of beta D galactose and beta D glucose. In this case, what actually happens is that here, uh, uh, first of all, let us describe a general concept that why do we consider drinking milk as healthy for our brain? Because inside our brain, the nerve cells of our neurons of our brain require sugar which is known as galactose. Galactose is that is why also known as a nerve sugar. Nerve sugar which provides energy to the brain in order to be consumed by the nerve cells in order to produce energy. That is why whenever we drink milk, it contains galactose, which is necessary for the nerve cells to work properly. That is why we consider galactose as a major source of uh, neurons energy. So accordingly, this is present here. That is why mostly people consider drinking milk. So here, galactose, uh, beta D galactose and beta D glucose are combined together with by the form of a linkage, which is collectively known as beta D galactopyranosyl 1 to 4 beta D glucopyranosyl linkage. So here, this linkage is a bit longer, but we don't need to memorize this linkage. We just need to go through the concept that what does this linkage indicate. This linkage indicate that here first we are having a beta D galactopyranosyl sugar. We are having beta D galactopyranosyl sugar, which is giving one of its which is giving hydroxyl group from its first carbon, which is giving hydroxyl group from its first carbon to the fourth carbon of hydroxyl group, due to which both of the hydroxyl group react to liberate the water molecule. On the other hand side, the fourth carbon atom of beta D glucopyranosyl is having its hydroxyl group from the fourth carbon atom with the first carbon atom of beta D galactopyranosyl sugar, due to which both of these hydroxyl groups are liberated and collectively form a bond between them, which is known as glycosidic linkage. Collectively, we don't need to memorize this, rather we need to go through the concept. First, we are having beta D galactopyranosyl sugar, and secondly, we are having beta D glucopyranosyl sugar. Beta D galactopyranosyl sugar is giving its hydroxyl group from first carbon, and beta D glucopyranosyl sugar is giving its hydroxyl group from the fourth carbon, due to which they form a linkage in between them, which is beta D galactopyranosyl 1 to 4 beta D glucopyranosyl linkage. So this is a long name, but you don't need to memorize it. Rather, you should go for the concept, because concept is what is required. So I guess that's all. And there's a simple difference between glucose and galactose sugar. What is the simple difference between glucose and galactose sugar? That here in simple sugar, for suppose in glucose and galactose sugar, in glucose sugar, basically the here we are having one, two, three, four, right? So here in glucose sugar, the fourth carbon is having its hydroxyl group on the right side, and fourth carbon, the glucose sugar is having its hydroxyl group over the right side, but in galactose it is having the hydroxyl group on the left side. This is the only difference between galactose and glucose sugar. I am not sure about number of carbon atom. Maybe it is fourth or fifth. So Well, it's wrong. Actually, there's a mistake inside it. So what I was saying is that the difference between glucose and galactose is such that in glucose we are having the second carbon according to the ring structure. The second carbon in glucose is having its hydroxyl group on the right side. The hydroxyl group is present on this side, but in the galactose structure, the hydroxyl group is present on this side, due to which we consider this structure uh, to be the only difference between the glucose and galactose. Rest all the structure remains constant. Just there's a minor difference between the second carbon. In second carbon, hydroxyl group is present on the upper side, and in the galactose and glucose, it is present on the downward side. So I guess there's one more mistake, that here we are not having the second carbon as this one, second carbon as this one, which I am writing here. This is the second carbon in which the hydroxyl group is present in the downward side. It is basically the structure of galactose, and the one which is having hydroxyl group on the upper side is known as glucose. And this is the structure of galactose. Hopefully you understood today's class. And the source of lactose. Source of lactose is sugar, is milk. Of course, major source of lactose, as it is predominantly found in the milk, lactose sugar, and sucrose is having its major source as cereal grain, tuber, cereal grain, tuber, as well as it is present in bulbs, fruits, vegetables, not in vegetables, but in cereal grains, tubers, bulbs, fruits. Yeah, and we can say that in legumes and some vegetables as well. So sucrose is present, and one more MCQ's point, that here sucrose. Sucrose is basically most abundant. Sucrose is most abundant disaccharide in the whole carbohydrate structure. So it is the most abundant disaccharide. Hopefully you enjoyed the video, and this is all about today's video.